So in the first segment on Freaks and Mutants, we talked about non-genetic reasons why our plants might look different, even if they're asexually propagated. In this mini lecture, we're going to talk about mutations specifically. So what is a mutation? Well, it's any change in the hereditary state of an organism. And this could be at the level of a single gene or a few genes, or it could be at the chromosome level, which is a more drastic change. All chromosomes have lots of genes on them, so if you're changing at the chromosome level, you're changing a lot of genes. When we get these genetic mutations, all kinds of things can happen. So if you look on the right here, this is a picture of a black-eyed Susan flower. Now, I showed you some regular black-eyed Susan flowers in the last mini lecture on one of the first few slides where phenotype was defined, so you can see what they usually look like. This one has a genetic mutation that makes the center of the flower stretch out and curve like that. It's a specific type of mutation called fasciation, but the bottom line here is mutations can really significantly change the look of your plant. So, for example, this flower may have been asexually propagated from a regular black-eyed Susan flower, but now, because of the mutation, it looks quite different. We have a few terms to refer to different types of mutations in plants. The first term is bud sport. A bud sport is a portion of a plant that looks phenotypically different from the rest of the plant. So that's what we can see in the picture here. We have a plant called coleus that this particular one typically has red and yellow leaves. You can see there's that one branch there near the middle of the picture with green and yellow leaves. That's a bud sport. Sometimes you'll hear them referred to in the trade as just sports. The next term is chimera. So this is a specific type of mutation in which the plant contains genetically dissimilar cells in different layers. So we're going to talk about that one in more detail. If you have a bud sport, you know that there's just some sort of mutation happening there. If you know it's a chimera, we know a little bit more in detail about what kind of mutations are going on. So another definition for chimeras is a genetic mosaic in which genetically dissimilar cells are present in the apical meristem. Remember that your apical meristem is that growing point at the tips of your branches or of your roots. In this case, we're typically talking about branches. Now, if you look at the picture here, this is a microscopic picture of a cross-section of a meristem. So you can see that looks a little bit like the cross-section of a bud. Well, actually, it is a cross-section of a bud. In that actively growing and dividing meristem, we have 3 layers, and we call them histogenic layers. They're just layers of tissue in the meristems of dicots. Monocots have a little bit of a different setup, but they also have histogenic layers. As the meristem actively grows and divides, these histogenic layers are going to develop into different types of tissue in the plant. So let's talk about what they develop into. The histogenic layers are named simply L1, L2, and L3 for layer 1, layer 2, and layer 3. So L1 develops into the outermost layer of the plant. So that involves the epidermis of stem, leaves, and flower petals. You can see I have a drawing of a leaf here, which roughly points to the tissues that each histogenic layer gives rise to. Now L2 gives rise to the sex cells of a plant, and also the inner tissue of lower petals, some inner tissue of leaves and roots, and some vascular tissue, so your xylem and phloem. Now, as you might imagine, if you get a mutation in L2, you're going to have some really weird stuff happening when you sexually propagate the plant because the sex cells have been mutated. And then finally, we have L3. This is the histogenic layer that gives rise to the centralmost tissues. It gives rise to some root tissue, some central tissue of the leaf, and the pith, which is a central tissue in the stem. When we have a chimera that we can physically see, it's usually in L1, the outermost layer, so that's where it's most visible. You might think of them like peanut butter M&Ms. So if we're thinking about it like that, L1 is the candy coating, L2 is the chocolate, and L3 is the peanut butter, right? If there's a L1 mutation, a mutation in the candy shell of your M&M, you're really going to notice as soon as you look at it. Now, there are 3 different types of chimeras. The chimeras can have mutations in L1, 2, or 3, or parts of 1, 2, or 3, or 1 and 2, or 2 and 3. You can have different combinations of which layers are mutated, but the three types of chimeras are sectorial, mericlinal, and periclinal. When you have a sectorial chimera, the mutated tissue encompasses part of all three histogenic layers. So it's not all of all three layers, but it's part of all three layers. So if you look at our picture here, if you imagine these little targets as L1, 2, and 3, the outermost ring being L1, the middle ring being L2, and the centralmost being L3, and then the dark portion is the mutated portion. So all of those pictures on row A have mutated tissue in L1, 2, and 3, but just certain portions. A mericlinal chimera has mutant tissue that encompasses part of only 1 or 2 of the 3 layers. And again, the shaded regions on row B would show examples of the mutated regions of various mericlinal chimeras. And then finally we have the periclinal chimera, in which mutant tissue encompasses all of 1 or 2 histogenic layers. Now, periclinal ones are the ones that we care most about for propagation because they're the most stable. You can see how in the sectorial and mericlinal chimeras it would be very easy, since these are actively growing and dividing areas of the plant, it would be easy for the normal non-mutated tissue to sort of take over by actively growing and dividing much more quickly than the mutated tissue. So for example, in the sectorial chimera on row A, the one on the far right, you only have a very small sliver of mutated tissue there. If your normal tissue continues to divide and grow more quickly and more substantially than that mutated tissue, then you might not be able to see any change at all due to that mutation because it's just such a small portion of the tissues. So again, periclinal chimeras are the ones we really care about for our purposes. They're easily propagated because they're highly stable. Since the entire layer or layers has mutated, there's not unmutated tissue in that layer that can mask the mutated tissue by growing faster. Periclinal chimeras can only be maintained in propagation via stem cuttings, division, grafting, and it should also say layering on that list. That doesn't mean that you can't propagate a periclinal chimera plant using, for example, leaf or root cuttings, but if you do that, the mutation will not be in the new plant. So, for example, thornless blackberries are a periclinal chimera. Blackberries in general can easily be propagated by root cuttings, but if you propagate your thornless blackberry by root cuttings, you'll get a bunch of thorny blackberries as the daughter plants. However, if you take a stem cutting of that thornless blackberry, you'll get more thornless blackberries. There are a lot of horticultural crops that are periclinal chimeras, particularly variegated ones like the variegated tulip poplar in the picture here. Usually that's a result of a mutation in L1, that outermost layer of tissue, and as a result of that, we get these variegated leaves. So sometimes the mutation is that L1 doesn't produce chlorophyll at all, or maybe not as much chlorophyll, giving it that lighter coloration. So here are some more examples of horticultural crops that are chimeras. Again, your thornless blackberry, which makes sense. That's still an L1 mutation because the thorns are on the outermost layer of tissue on that plant. All right, so thornless blackberries and raspberries. Marble type poinsettias, like the pink one shown here, it's very similar to what you get with leaf variegation in a marble type poinsettia, so those are typically chimeras. And then I put 2 examples down here at the bottom. These 2 plants are the same species, Daphne burkwoodii, but they're 2 different cultivars. The one on the left is a cultivar called Carol Mackey. Carol Mackey is a chimera. It was a chimeric mutation that someone discovered. They propagated it by stem cuttings, and now they have this new variegated cultivar. Usually Daphne burkwoodii just has solid green leaves. Then, as someone was growing lots of Carol Mackey, one day they noticed a bud sport on it that looked like the plant you see in the right there. So they propagated that bud sport and they came out with a new cultivar called Briggs Moonlight. So that's just an example for you of how these mutations can give rise to new plant cultivars. Now you can see how these sort of mutations can be beneficial for the horticulture industry, and so you can actually induce mutations in a plant on purpose. There's a couple of ways to do this. One is to expose the seeds of the plant to extreme temperatures or to store them for a really long time. And when I say a really long time, I mean like decades. Also, growing plants in tissue culture, just the tissue culture environment, and we'll talk about this more later when we get to tissue culture, but the tissue culture environment just tends to encourage mutation. So sometimes just by propagating a plant in tissue culture, you'll get more mutations. But you'll see that the last two bullet points are highlighted there. These are the two methods most commonly used by plant breeders. They're more reliable. And that's radiation and chemical mutagens. You either radiate the plant or you expose it to chemical mutagens and then propagate it to see if you can get any favorable mutations from those treatments. Sometimes it works, sometimes it kills the plant, sometimes it doesn't kill the plant and the plant doesn't show any mutations at all. This was something that I actually did as part of my master's research. We grew them in tissue culture first because it's easier and safer to expose tiny plants in tissue culture to radiation or chemical mutagens than it is a whole big potted plant. But we exposed the plant to these things. Nothing happened. It looked like you hadn't exposed it to any horrible chemicals or radiation at all. It just looked like normal. But sometimes it works, and if it does, you might get a mutation that has a favorable appearance or some kind of favorable trait, and then you can make that as a new cultivar. So with that, I'm going to end this mini lecture. The last segment will be about pathogens that affect asexually propagated plants and make them look different from each other.